Hooked on a feeling. I'm high on the.
Against the constant threat of mutation, only a few proteins prevent absolutely everything from going wrong.
Cancer develops from the accumulation of genetic and epigenetic alterations which can activate oncogenes or may inactivate tumor suppressor genes. Knudson's two-hit hypothesis states that TSGs are genetic material that suppress tumor-like properties and act recessively, meaning both genes are to be inactivated for the loss of growth controls. The cancers that arise may be hereditary or sporadic.
The RB gene is a major inhibitor of the cell cycle. The RB gene is active when it is hypophosphorylated and bound to E2F, which inhibits transcription. Otherwise, it is inactive when hyperphosphorylated and releases the E2F to continue transcription. RB is a rare cancer occurring in young children, with the majority of cases being familial retinoblastomas. Sporadic RB1 gene mutations can result in bone, lung, and breast cancers.
Adenomatoous polyposis coli gene functions in cell division and adhesion by regulating the beta-catenin activity and the WNT pathway. Germline mutations of the APC gene result in familial polyposis syndrome, which are polyps affecting the colon, which may become malignant. Otherwise, sporadic mutations lead to other cancers, such as colorectal cancer. A majority of these mutations are resultants for frameshifts to deactivate the protein function.
BRCA1 and 2 are another tumor suppressor gene whose function is to repair damaged DNA.
That and they ask you how you are, you just have to say that you're fine when you're not really fine, but you just can't get into it because they would never understand.
BRCA genes are highly responsible for breast and ovarian cancers due to germline mutations or sporadic mutations by methylation.
DNA mismatch repair genes such as MLH1, MSH2, MSH6 and PMS2 are genes responsible for repairing DNA mismatches during cell replication.
Hello, darkness, my old friend.
Inactivation of MMR genes result in microsatellite instability. Germline mutations of MMR genes result in Lynch syndrome, which is a hereditary, non-polyposis colorectal and endometrial cancer.
TP53 is a crucial protein involved in cell cycle arrest, DNA repair and apoptosis, and is a nuclear phosphoprotein that targets specific genes and acts as a transcriptional regulator. TP53 is a protein bound to MDM2 that remains inactive, but with a buildup of stress, TP53 becomes phosphorylated and activated by the inhibition of MDM2. TP53 is the most common gene mutated in cancers. TP53 germline mutations have a higher cancer risk of Li-Fraumeni syndrome, which increases an individual's risk of developing multiple cancers and multiple tumors. Besides just mutations of the TP53 gene, it may also be inactivated by binding to viral proteins such as simian vacuolating virus 40, adenoviruses, and human papillomavirus.